This is the first public presentation for those who don't know of him, of the process. Next, after that will be his proposal defense, his actual dissertation defense. Um, so he's going to present today. Let me ask him, is his committee here?
Um, it looks like we might still be waiting on Shannon.
No, I see her.
Oh, is she here? Okay.
Yeah.
And then is Jubo here? I see going Ming.
Yeah, Shubo's here.
Okay, perfect. Then
Good.
everyone's here already.
Okay, great. Okay, so I will give a brief introduction and not take too much of his time. Nate is known to many of us in the department. He's a PhD candidate in the bioinformatics and computational biomedicine major. He originally studied biological physics at the University of Washington and then worked as a mechanical engineer developing a lung cancer diagnostic device using 3D single cell imaging. His primary research goals are to develop robust drug response models to aid decision making in precision oncology. And his current work has broad translational application in drug development, combination therapy prediction, clinical patient response prediction, and patient therapy toxicity prediction. Several of us, many of us maybe actually heard a wonderful talk a couple weeks ago at the AMIA Symposium by Josh Denny on precision medicine. And so we're attuned to this topic. So again, he's going to present the current status of his research at the symposium today, and we'll get feedback from us as he develops his proposal. So I will turn it over to Nate.
Thanks, Bill. That was a great intro. Sounds like some of my background is already taken care of. All right. So is everyone seeing my slideshow here?
Yes, yeah, we see it fine.
Excellent. Okay. So, sorry, seem to have a screen in my screen. There we go. Okay, so like Bill said, my name is Nate Evans, and today I'm going to be giving my symposium presentation. So the title that I've given this is Improving Uncertainty Quantification in Drug Response to Guide Precision Oncology. And so a little bit of motivation for my research. Since coming to OHSU 2 years ago, I've worked in a number of different labs with Shannon McQueenie in the BioDev lab and Dr. Coles Martin, as well as more recently with Dr. Gordon Mills. And all of these labs have a lot of focus on precision oncology tasks, which have really guided my interests and the work that I'm going to present here today. But also, you know, my interest really is in, like Bill said, developing these drug response models and how we can use those for combination therapy prediction and drug research prioritization. So, quick background on the talk today. I'll start with a brief overview of precision oncology and some background. And then I'm going to spend the first part of the talk trying to show the connections between decision utility, a concept called model calibration, and then finally uncertainty quantification. And then from there, we'll move on to applications of uncertainty quantification in drug response. And then we'll finish up with some of the areas that I'm most interested in pursuing, kind of a teaser for my proposal. So precision oncology, if I think it's becoming more and more popular these days, but it's a bit of a shift away from a one drug fits all style of medicine, and it operates under the assumption that there's heterogeneity in patient and tumor genomics, and this is going to lead to variation in therapeutic response. And so the goals of precision oncology then is to use omic profiling or some kind of feature, patient or tumor feature, to try to stratify these patients for in a way that can improve treatment. So we want to choose the optimal therapy based on those tumor characteristics or the patient characteristics. And there's huge potential here, definitely to improve patient outcome. That's the primary goal. But also there's significant work being done to reduce toxicities and side effects. And then finally, in the long run, we also think this might be able to reduce financial burden, but that's kind of a long-term goal. And so to get to the kind of the potential of precision oncology requires a lot of research, and there's still some pretty significant bottlenecks. So in this figure at right, it really identifies a lot of these issues where we start out with a ton of drugs, and through the development process, there's a significant attrition rate. And so to go from a brand new drug to drug discovery to FDA approved drug can take 10 to 20 years, and by some estimates around $2 to $3 billion. And so this is a huge obstacle, and we want to try to improve this. There's a lot of reasons why this is such an issue, and we're actually going to talk a lot more about those sources of uncertainty and why we see such high drug attrition rates throughout this talk. So another task in precision oncology that's gained a lot of effort in the last few years, or the last 10 years, is this idea of drug combination therapy. And there's a number of reasons why this is valuable. Number one is that when you use monotherapies, oftentimes we see good initial response followed by either tumor resistance or metastases. And we think this is due to tumor heterogeneity. And so if we have multiple cancer clones in a tumor, then a single monotherapy often acts as a selective pressure, and you get evolved resistance over time. And so one approach to this is that by using combination therapies, we may be able to reduce or improve the durability of response and prevent that tumor resistance or metastases. Another concept here is that there's often multiple driver genes in a cancer, and so by targeting all of those or multiple of those driver genes, we may be able to improve efficacy of that therapy. There's also a concept of drug synergy, where the response to a combination therapy is much better than would be predicted by either of the single monotherapies together, and this has a lot of value in reducing toxicity. It can also improve the therapeutic window. And so when we try to find these drug combinations, it's usually done through high throughput screening with dose response assays. But what I want to draw attention to on the right here is that this is a fairly complicated or very large search space. And so on the x-axis, we have the number of drugs we're choosing from, and on the y-axis in log 10, we have the number of possible combinations. And this is a fairly simple combinatorics problem, but you can see that we have very high exponential growth, and depending on how many drug combinations you're looking at, this becomes incredibly problematic. And just to provide a little context, the Broad Repurposing Hub, which is a major repository for information related to drug repurposing or combinations, has over 6,000 drugs included. So that search space is incredibly large. And when you add the genomic axis or the omic axis here, it becomes even more complex. And that kind of brings us to the role of drug response prediction. There's a number of reasons why we use drug response models in precision oncology. As I kind of alluded to, we want to use them to prioritize different therapies for high throughput screening or for further research. There's also some really fantastic work being done in chemotherapeutic choices for patients. So in breast cancer, there's two screens, Oncotype DX and MammaPrint, that are both used to help physicians choose which therapy is going to be most valuable. And then in research settings, we definitely want to use our drug response models to understand the underlying biology. And so there's a lot of value there. And then lastly, in today's research settings, we have a huge amount of data, all kinds of different sequencing technologies coming out. and we can produce an incredible number of features for a given tumor. But the issue is that us humans aren't great at integrating that all together and using it in any actionable way. And so that's another fantastic role that these statistical models can try to fill. And then, so just a little more context about the talk. I'm going to be primarily talking about cell line models. And so when we build a statistical model for cell lines or any model system, we're usually using dose response curves to measure that functional response. And that's characterized by two metrics, either the area under the dose response curve, the AUC, or the inhibitory or the 50% inhibitory constant, IC50. And so I'll probably be referring to those throughout the talk. And then inputs are commonly bulk sequencing of monoclonal cultures, and we'll talk a little more about how we encode drugs. There's a number of ways to do that. So, as everyone would probably expect, there's a ton of research being done here. There's a lot of different incredible models that have already been developed. This drug cell was published October of this year, and it's actually a really incredible paper if anyone has interest in this field. But I think it represents, state-of-the-art drug response predictions. And what I want to show here is the model on right. The x-axis is all the different drug response models or predictive value of the drugs. And then on the y-axis, we have the performance, and they use Spearman correlation there. And so in red on left, these are pretty high-performing model or high-performing drugs that are quite predictable. But the blue or purple on right, these are not as well performing, and we recognize that there's a huge number of these. So this really represents a lot of room for improvement and work still to be done. And there's a number of reasons why we still have such variable response. This is probably what I think about most often, that the data types that we use in these models are incredibly noisy. And so what the graph on right is comparing three of the primary public pharmacological data sets, CCLE, GDSC, and CTRP. And so, and what it's looking at is for a given cell line and drug combination between these three data sets, how close do those values correlate? And so the correlation is on the y-axis there. And you can see that except for these adjusted metrics, almost none of the metrics have a correlation above 0.6. And for context, that last model I showed you, they used CTRP and GDSC. And so if we're building drug response models using these data types, we're going to be fundamentally limited by this noise or this poor concordance. And there's a number of reasons why that is, which we'll get into even more later on. So there's kind of two ways to approach this. The first is that we want to understand where we have uncertainty in drug response data types and improve upon that. And that's that's definitely something that we all want to work on. But the second way is to say, even if we have moderate performing drug response models, can we use these predictions in an actionable way? And that brought me to uncertainty quantification. And these are some papers that kind of echo my thoughts that we really need to start incorporating this. So to understand what uncertainty quantification is, it's first, I first need to introduce this idea of calibration. And so when we think of model performance, I think a lot of us, at least, or at least myself, initially thought of discrimination. And so that's the ability to predict a correct value. And the metrics for this are accuracy and sensitivity specificity, or AUROC. But there's kind of another branch to model performance, which is the ability to assess our uncertainty in the predicted value. And this is what we call calibration. There's a number of metrics we can use, especially in classification problems. but we'll look at calibration plots today. So a calibration plot, it attempts to evaluate how well a model characterizes its uncertainty. And so on the x-axis, we have the predicted probability of some observation. And then on the y-axis, we have the true probability. And this is to implement this, it's usually done using bins. So for instance, for a model between, or for any model, you would take all predictions between probability 0.5 and 0.6, and you would ask how many of those had a positive. And so for this model characterized by the blue line, it has almost 0.85. So that's really quite high. And what we would like to see, a perfect model or a well-calibrated model, would lie right along the diagonal, as that would encode the uncertainty properly. One other aspect to consider when thinking about these calibration plots is the distribution of probabilities that are predicted. And so in this example, we can see that probabilities between 0.9 and 1 are almost never predicted. And so the majority kind of lie in these two humps around 0.3 and 0.7. And that really is another factor in actionability of predictions here. So as a quick example of why these calibration plots are useful, it allows us to kind of discretize model performance, and especially in terms of probability. And so for that red line right, we can say that around 10% of the observations have predicted probabilities between 0.5 and 0.6. And of these, 85% of the observations are correct. And so for this model, it actually significantly, it's significantly underconfident. It underrepresents the predicted probabilities between 0.5 and 0.6. And another kind of interesting aspect of why these curves are useful is in the same way that we can characterize humans as confident or underconfident, and that affects our ability to trust them. We can kind of do the same thing with these predictive models. So this upper one represents kind of an overconfident, and the lower curve represents underconfident. And just to provide a human example of this, my fashion sense is very overconfident. So, something to be aware of. There's a number of reasons why we should care about calibration. It's really important when we want to use our predictions for decision making, and I'll show why. But even more so if there are costs associated with predictions. And then probably most applicable to my research, it can be very useful if our models have poor discrimination performance, and that could be due to noisy data types or perhaps low data volume. I also really like this example. I think it shows, it's useful for showing why calibration is something we should pursue. So in a lot of clinical and research settings, there are already decision-making processes in place. And so this kind of lets us incorporate model predictions on a case-to-case basis. And the advantage of this is that we can mitigate risk by only letting a model incorporate its predictions when it's very confident. And in a well-calibrated model, that means that we can use the tails of this distribution where it's quite confident down here and quite confident up here, and only use those predictions and kind of disregard the region where we're uncertain about prediction value. But it's critical here that our model calibration, that we have good model calibration. Okay, so next, I want to show the relationship between model calibration and decision utility. And so to do that, I'm going to introduce these decision curves. They were originally developed to evaluate clinical decision utility of a predictive model. And but I'm going to try to explain why they're useful in general. So on the x-axis, we have you can think about this as preference or consequence. And it's in terms of threshold probability. So what threshold do we use to choose an intervention or a positive, essentially? And then on the y-axis, we have net benefit to the process that we're interested in. And we compare against baselines. So this slanted black line is intervention for all, and then this bottom line is intervention for none. And so the difference between our model curve in light gray and those baselines is essentially improvement on the decision process. And if you do have another model or a diagnostic test, you may be comparing against those baselines. And so there was this great paper, and what they did is they compared 3 models with identical discrimination performance, but then they varied the calibration of the model. And what they showed is that this first line, the light gray, can people see my pointer? I hope so. The light gray here.
Yes.
Okay, thank you. Is a perfect calibration. But as the calibration gets worse, that decision utility also gets significantly worse. And even more so, we can notice that it's actually worse than no intervention at all. So there's negative contribution to this, this decision process. And that's definitely something we want to be, pretty aware of if we're developing a model that has any kind of input into a decision process. This calibration becomes quite valuable. Okay, so that was a lot of background, but putting it together, I hope that I've shown that in this drug response field, we have quite variable predictions, and there's a lot of room for improvement. And one of the concepts here is that model calibration has been, in a lot of cases, overlooked, I think, in this field, and we can improve the actionability of some of these drug response predictions if we can also improve those model calibration. So the next question is, how can we improve model calibration? And that brings us finally to uncertainty quantification. So in a machine learning context, we traditionally use scalar metrics as or scalar variables and as inputs and outputs. And then moreover, our parameters of the model, if we have a parametric model, are usually scalars as well. And what this does is it collapses any uncertainty information that we might have in inputs or outputs or the model parameters. And so our goal here is to start thinking about how can we include that uncertainty. So we want to use distributions as our input variables or as our output variables. And then there's also methods that we can use distributions to characterize our parameter models. And what that does is it allows us to propagate uncertainty from any of these sources into predictions, into novel predictions. And so there's a number of ways to do this. We can sample from input variables and propagate that through to prediction uncertainty. There are also ways to try to propagate our inverse, propagate our output variation back through model during training. And then finally, there are some methods that kind of implicitly characterize uncertainty. So real briefly, I'm not going to go into depth on these methods, but there are a number that do implicitly characterize uncertainty quantification. You can do this to some extent in frequentist methods, and for instance, most classification tasks have some element of uncertainty. But to improve on this, the Bayesian field has really produced a lot of fantastic methods, and I'd say the majority of the work has stemmed from there. Gaussian processes are probably one of the go-to methods. Bayesian regression can do similar things. However, it's usually parametric. And then you can also do Bayesian neural networks. And then more recently, in the last 10 years or so, deep learning has put a lot of effort into how to use probabilistic models. So variational autoencoders can do a good job of that. There's been some work in using, so dropout is a mechanism usually used for regularization. However, if you use dropout during prediction, then you can produce essentially an ensemble of predictions and quantify uncertainty. This has been shown to approximate a number of Bayesian inference methods. And then finally, if you set up your prediction task as an interval task rather than a single value, you can also quantify uncertainty that way. In general, you can break down uncertainty into two broad categories. The first being aleatoric uncertainty, which we can think of as our irreducible error. And on the left there, we have high aleatoric uncertainty. And on the right, we're going to have low uncertainty. And then the other form is epistemic uncertainty, and that's where we just don't have enough data to characterize the model at all. And both of these can contribute to uncertainty in predictions. It can be quite difficult to separate the uncertainty in some cases. So before we go into types of uncertainty or sources of uncertainty in drug response, I just wanted to give a little context in if we do uncertainty quantification well, what methods or algorithms, essentially what are the applications for this? And so one of the ones that I'm most interested in is task or cost optimization. And there's a number of algorithms that use the upper confidence bounds to try to optimize some given function. This is Bayesian optimization is one of those. There's also Bandit algorithm. As we've talked about a little bit already, there's a lot of application and decision utility through improving calibration. There's been some examples where they were able to improve analysis by quantifying that uncertainty. This is a really interesting concept where if we include uncertainty in our interpretation mechanisms, then we add this new axis of, we believe that that this prediction was made for this reason, but we have some level of confidence on it. It's also been shown that uncertainty quantification can affect, so if you provide a confidence metric to a human user, that affects how likely they are to use the prediction for some task. And then finally, there's applications in risk mitigation. And then in precision oncology, these have pretty broad application to combination therapy prediction, to drug repurposing, and then certainly in developing clinically actionable models or for patient stratification. Okay, so for the next 10 or 15 minutes, I'm going to go over where we find uncertainty, what the sources are of uncertainty in drug response. So in general, we can have uncertainty in any of our inputs or in our outputs. And then we also need to recognize that most of my work is in model systems. And so we develop these predictive models using cell lines, or in some cases, we might use mouse models. But they're really only valuable if they predict patient tumor response in some way. And so that external validity becomes very important. And we know that there's a lot of reasons that our model systems don't always agree with patient response. And then one other thing, as I go through this, recognize that I'm trying to explain why there's variation or uncertainty in this field, and what's been done to address it so far. But if we can't address it or needs more work there, then we want to think about how can we quantify the uncertainty to improve the actionability of those predictions. Kind of two sides there. Okay, so drug encoding is important as that input variable for drug perturbation. There's a number of ways to do it. Probably the most naive way would just be use factors. However, there's also been success using molecular structures. The encoding that I'm most interested in is thinking about it as a protein target, right? And so to understand that, we have to think about the pharmacological models of protein ligand binding. And this at right here is a simplified version of this, where we have an inactive protein, and when the drug binds, it stabilizes the active conformation and produces some downstream effect. And then what's really important to know in this scenario is the proportion of unbound or free proteins and drug to the bound complex. And that's characterized by our dissociation constant, which we sometimes call the binding affinity. And so if we know which proteins a drug binds to and the binding affinity for that given drug protein, then we can characterize a drug's perturbation on a model system. And I think that the pathway provides a lot of context in this. So this is the MAPK pathway, and we can think about, a lot of these systems have a receptor tyrosine kinase and then a signaling cascade that ends in transcription factors that lead to some measurable change. And so encoding our drugs as these protein targets, we can think of disrupting that pathway or that signaling cascade to try to change cell behavior. Another issue in this drug encoding, when we're thinking about it as protein targets, is we really need to know all the targets that a protein might affect. And this can be problematic because there are known and unknown targets depending on how well studied a drug is. So imatinib, for instance, is a fairly well studied drug. It was developed to treat chronic myeloid leukemia, specifically to target ABL1 originally, but through more research, we found that it had a lot of secondary targets. So all these different colors represent different targets. And this plot shows the level of evidence for different binding affinities of those targets. And so we can see that we have kind of inconsistent drug data, and there's significant uncertainty there. Moreover, if we look at other pharmacological constants, we see additional variance. And so this can be really difficult if we want to encode drugs in this way. It can be difficult to know all of the protein targets. So another important factor here is that different drugs can create different effects in a system. And so that model that I described earlier, that would be a agonist, where it binds to the protein and creates some downstream effect. However, kinase inhibitors, which are a really common class of drug that we use in precision oncology, would probably be considered an antagonist or potentially an inverse agonist, where it binds to a protein or a kinase and it stabilizes the inactive conformation, preventing signaling. And so recognizing that different drugs will behave different ways and that we need to understand that mechanism of action if we're going to encode them in a reliable way is actually quite important. And that's information we don't always have. Okay, so moving on to our second input variable, which is our omic encodings. I like to start with this concept that we really have to deal with surrogate variables, and that's kind of shown or can be shown with the paradigm concept. And so paradigm was this pathway modeling algorithm developed in 2010. And they did a great job of incorporating the central dogma of biology in a factor graph. And so they were able to encode, DNA genomics in this left node, and then RNA expression. If we had protein expression, we might be able to include that with the goal of trying to model protein activity. And what's really important to recognize is that a drug response really lives in this right node in protein activity. And so we're trying to understand that, but we have some separation with the variables that we're actually measuring. And for anyone who doesn't work with this stuff, RNA expression and DNA mutation is probably, it's just far easier to measure those than it is to measure protein or protein activity. And so we definitely have that separation there. And that can result in quite a bit of uncertainty. As a quick side note, there was this fantastic paper developed in 2016 where this was from the Califano lab. And they took a bit of a divergent approach where they said, rather than looking upstream of protein activity, let's look downstream. And if we know genes that are regulated by a given protein, we can try to infer that protein's activity levels. And this algorithm has seen really, really incredible results. So I'm excited to see where that goes. Another source of uncertainty in these in the genomic encoding is either heterogeneity in our cells or the lack of variation that we're able to measure. And so in my work, we majority work with monoculture cell lines and measure the genomics using bulk sequencing. And what's important to recognize is that bulk sequencing is an average of all our cells. And so if we have any variation due to, it could just be biological variance like transcriptional bursting, or if there's any transient cell states, then we're going to collapse that into a single value, and that can be problematic. So this plot at the bottom kind of shows how if there is variance, we collapse that into a single point, and this becomes far more problematic if you think of a marginal distribution where there might be complex or bimodal distributions. One other aspect to consider is there has been some work in deconvolving bulk sequencing into individual signals. I believe this is more appropriate if you know which cell types might be included, and so it's kind of dubious whether it'd be valuable to monocultures, but it's definitely some valid work in the field. Okay, so moving on to our output variables, drug response. Like I said before, drug responses in cell lines are usually characterized by dose response curves. And since around 2012, there's actually been a number of papers that showed a growth rate bias, which kind of confounded a lot of these. And then more recently, the Sorger lab put out this paper where they showed really definitively that important metrics, summary metrics like IC50 that we talked about before, and Emax, which is another important parameter that characterize these dose response curves, are highly correlated with cell division rate. And so, a faster a cell grows, the lower the IC50, or the higher the IC50 rate is. And so this is a problem because when we use these metrics in drug response, we want them to encode drug response, not cell division rate. That's a separate piece of information. And so the point is that we have quite a bit of uncertainty in these classical drug response metrics. There has been some really great work. This was also done by the Sorger lab, where they developed a new metric. They called it GR, which stands for growth rate normalization. And in simulations, they were able to show that this GR metric is robust to cell proliferation rate, whereas IC50 or EC50 are not. So that's a significant improvement in the field. There's also a number of other reasons why we see a lot of variation in drug response. Experimental conditions can be have a massive factor. And then there's also processing components. Another concept here is that we currently don't do a great job encoding, dealing with the time axis of drug response. So when we measure a cell line's response to a drug, we usually do it at a single time point. It might be 48 hours or it might be 90 hours. But there's also been work. So this plot at right is showing one cell line's response to the drug omeprazole. And what you can see is that the GR50, that metric that characterizes the drug response, changes depending on when you measure it. And it changes pretty significantly. And so if you measure this at a discrete time point, say 48 hours, you're going to get a very different value than if you measure it at 90 hours. It should be noted that I suspect this was a bit of a cherry-picked example, that not all drugs and cell lines will behave this way. A lot are far more stable. But the fact that this is a possibility really, contributes to uncertainty in our drug response. And finally, we have uncertainty in that external validity. So like I talked about before, we are working in model systems to build these drug response models. And we do that because we really can't produce enough data in humans, or we can't measure what we want to measure in humans. And so we really have to work in model systems. But the goal is that we want these model systems to be representative of the human. And there's a lot of issues with this. For instance, we know we're going to lack immune response in both cell lines and mouse models. The pharmacodynamics and the pharmacokinetics will be quite different. And then in a lot of cases, so in mouse models, you might be able to recreate a microenvironment to some extent, but in cell lines, you won't be able to do that. And then there's also this growing work in tumor heterogeneity that we know contribute pretty significantly to drug response. And that's something that can be very difficult to do in these simple model systems, at least. Okay, so before I wrap up, I wanted to give a bit of a teaser for my proposal. I hope I've kind of shown the relationship between calibration and uncertainty quantification and how that can improve decision utility, and then a number of sources of uncertainty in drug response. But there's kind of three primary areas that I would like to kind of address moving forward. One, I think there's a lot of room for improvement in quantifying uncertainty in our drug response metrics. So those the issues we see with IC50 or AUC. And I think we can improve this by trying to use probabilistic models to fit dose response curves and then propagate the uncertainty onto those summary metrics. And by doing that, we can improve model calibration. The second avenue here is, I think there's some pretty clever things we can do in encoding drugs as edge modifiers in a protein-protein interaction network. By doing this, it gives us a really flexible and mechanistic way of encoding these drugs. And then finally, this culminates in a bit of a complex idea, but Bayesian optimization is often used to optimize some cost-intensive task. For instance, they do it for hyperparameter optimization sometimes in machine learning. But it's also been shown to work well for drug development or chemical design. And the idea is that if you can build a continuous distributed representation of that represents your system, so this would be a one-dimensional version of that, and then you have some metric that characterizes how characterizes the response to the system, whether it's a good drug or a bad drug, then you can try to use these upper confidence bounds to suggest regions that are going to be more effective than other regions. And so I want to use that to to prioritize drug combinations. And I'll be talking a lot more about that next quarter, I hope. So in summary, we can improve model calibration by improving decision utility. We can, that if we do this, if we improve model calibration, we can improve, that by using uncertainty quantification, we can improve model calibration. We're kind of limited by these noisy data types in precision oncology, and that the huge search space required for combination therapeutics is really predicates that we need some prioritization methods. I hope I've gone over kind of a broad strokes on sources of uncertainty we have in this field. And then finally, UQ can really enable powerful algorithms and that are broadly applicable to precision oncology. So with that, I will acknowledge the DeepMice department and staff and all the students that helped me get here, the Colas Martin Lab for their help, and then the Mills Lab as well more recently, and then finally my advisor team, Dr. Aurora Blitzer and Dr. Shannon McQueenie. And I'll take any questions.
Great. Thanks, Nate, for an interesting talk. I guess one question I have as someone who kind of lives more in the clinical world and less in the weeds of the models that you presented. Have these approaches achieved success in repurposing drugs? Are there examples that can be pointed to that some aspect of this has led to some therapeutic advancement?
Yeah. So, are you talking about this last aspect or just uncertainty quantification in general?
Well, I'm really talking about the big picture. So, there's as every clinical person knows, there are many diseases that could use better therapeutics, and I'm just wondering if this repurposing approach, which is very logical, has actually resulted in benefit. Some drug being repurposed for cancer, or COVID-19, or any other condition.
I don't have any explicit examples of a drug that's been effectively prioritized, but I can say, so drug cell, one of those, that model that I introduced really early, that's actually seen a lot of that it's really valuable for prioritizing combination therapies. And I think just in general, it's due to that intractable search space, we're always trying to use these predictive models to improve, to guide our research efforts.
Good. Okay. There's a question in the chat, and others can feel free to either unmute or type into the chat. There's a question from Ted on, are you going to have a clinician on your committee? Somewhat related to the question I just asked.
Yeah, that's a great question. I would like to. I haven't settled on a committee yet, but I think that would be a really valuable contribution. Yeah.
He is working. This is Shannon. He is working though Gordon Mills is a clinician, right? So he has been working more closely with Gordon and actually been doing work in his lab as well. So he is getting some exposure to that.
Yeah, good. Any other questions?
Nate, this is Guami. It's a really great talk. So I have several questions as a member of your committee. Okay. So do you have actual a definition about uncertainty, since the whole theme about in your symposium about uncertainty, what we can really assign some kind of factors or some kind of things into uncertainty. Otherwise, it seems to be, especially the last 10 slides, seems like uncertainty is everywhere So, but then statistics, as you see, basically is trying to really handle all this uncertainty, right?
Mm-hmm.
Yeah, so then we have statistics already available, like all your models, actually, we can basically just plug in a probabilistic terms in all like a prediction model, even ordinary differential equation models, we can plug in probabilist terms. Then automatically convert all these models into some kind of like you see or uncertainty quantification. So my question, if make it short, is do you have actual definition about this uncertainty?
Yeah, that's a great point that, statistical methods, they all try to do this anyways. So when I say uncertainty quantification, the goal is really to be using distribution forms of our inputs and outputs and those model parameters, and in a lot of cases we collapse that, even in statistics, and that, in my opinion, it limits how much information which is relevant to model calibration, and limits that. And so I would focus on uncertainty as what can be encoded in a distribution form rather than a scalar metric. It's not a fantastic definition, but yeah.
Okay. So another question is, so about this model performance evaluation. So basically, we have classical like discrimination method using sensitivity, specificity, AUC, all this kind of thing. And you think we have the thing you actually, this is first time I heard about this calibration. So what exactly differs? Like from one left-hand side, can we actually try to do also calibration? In other words, can we use the same data set to do both discrimination and calibration for model performance evaluation?
Yeah. So you could evaluate both discrimination and calibration on the same test set, the same holdout test set. But it does require the same machine learning principles of creating a holdout test set or doing cross-validation.
Okay, so are there some kind of difference requirement for the actual data set for doing these two different calibration, two different evaluation?
Not that I know of. It's definitely some different metrics, different ways you think about the performance. But as for evaluation, I think you can treat them fairly similarly.
So for example, if we have a classifier as a machine learning model, so for discrimination based, basically we can have only true or false. But then can we do calibration since we cannot really see the actual probability in the different discrete bins? If you go to the next slide, so you basically
I see.
you need to know all these kind of scores, right?
Right. So if your model predicted only binary values, I don't think you'd be able to, you would not be able to evaluate model calibration. But in most in a lot of classification tasks, I think that we usually predict probabilities and then use a threshold to binarize those further. And so if you're dealing with those predicted probabilities, you could still characterize this calibration. I might have missed this point, but you can also do model calibration with regression tasks. You just have to use the quantiles in a little bit different of a way, but that almost all the work still applies.
Okay. That's interesting. I guess it's very difficult really to get the golden standard, the golden data set to really use it as a reference to do calibration, right? Since you need to have all these different scores. Or actually, you get the scores, especially
Right.
like in a classify environment.
Right, right. And one of the reasons that I suspect calibration has been kind of overlooked in the past is that if you have really fantastic discrimination, then this becomes somewhat irrelevant. If you're working in 95% accuracy range, then it becomes less important to be thinking about calibration. However, if you have moderate or poor performing models, then calibration becomes absolutely critical to using it in an actionable way.
Okay, thank you. I should have give other people some minutes.
Hey, Nate, this is Ben.
Hey, Ben.
I have a question too. Really enjoyed the talk. I just wanted to ask for a clarification on one of the things you mentioned regarding when that model outputs a scalar. If the model were a neural network and you were using dropout to do the kind of approximate the Bayesian for the calibration, you could still have a scalar output, and wouldn't you just resample it based off of a static input while applying dropout? Is that correct?
Yeah, so that's that is one way that they've tried to implement these uncertainty quantification methods. I think they sometimes call that Monte Carlo dropout or dropout at prediction, and yeah, it was able to approximate a number of Bayesian inference methods using that.
Okay, cool. That's really interesting.
Yeah, it's pretty clever, huh?
Yeah. Cool. Well, thanks.
There's another question in the chat from Rory. Can you see it, Nate?
Yeah. So Rory asked, do you have some thoughts on how you might approach handling concordance or disagreement of data types? For instance, if I'm using RNA or protein data. Yeah, that's a great point. I suppose in that example, so it really depends on what we want to infer. So in that central dogma, model, protein is going to be far closer to protein activity than our RNA. So I would probably weight it, I would weight protein more heavily. But that's definitely, definitely an interesting concept. I'll have to think about that one a little more.
Any other questions? If not, we'll look forward to your coming back and defending your dissertation proposal and see which direction you want to head and go from there.
Thank you. I appreciate everyone watching and the help, Bill.
Great. Thanks, everyone, and we'll see you at the next conference, which I believe is next week. Bye-bye.